We will now look at Week 5, Patterns of Single Gene Inheritance, Part 2. Single gene inheritance refers to the modes of inheritance which are the patterns in which single gene traits and disorders occur in families. Single gene disorders are determined primarily by the alleles at a single locus. They follow one of the classic inheritance patterns: autosomal recessive, autosomal dominant, X-linked, and Y-linked. We will now look at phenotype versus genotype. The phenotype is the expression of a gene in traits or symptoms, and a genotype is the allele combinations in an individual that causes traits or diseases. We will now look at Gregor Mendel. Gregor Mendel was the son of a farmer and the grandson of a gardener. He learned early how to tend fruit trees. At age 10, he left home to attend a special school for bright students, supporting himself by tutoring. He eventually became a priest in a monastery where the priests were teachers and did research in natural science. Here, Mendel learned how to artificially pollinate crop plants to control their breeding. As a university student, he became interested in applying statistics to plant breeding, and he thought about a question that had confounded other plant breeders. Why did certain traits disappear in one generation, yet reappear in the next? He had no knowledge of DNA, cells, or chromosomes. His laws of inheritance explained trait transmission in diploid species. He was able to describe the units of inheritance and how they passed from generation to generation. And he conducted experiments from 1857 to 1863 on traits in 24,034 plants. In looking at Mendel's experiments, he followed single traits with two expressions, such as short and tall. Short with short, tall with tall, and tall with short, resulting in hybrids. He also self-crossed plants. Mendel was able to deduce that consistent ratios of traits in the offspring indicated that plants transmitted distinct units. He analyzed genetic crosses of peas. P1 was the parental generation, F1 was the first filial generation, and F2 was the second filial generation. What you can see in this slide here are the traits that Mendel studied. He studied the transmission of 7 traits in the pea plant. Each trait had 2 easily distinguished expressions, or phenotypes. In Mendel's experiments, the true breeding was where the offspring had the same trait as a parent. An example is short parents produce all short offspring. The observed trait is dominant. The masked trait is recessive. Mendel conducted up to 70 self-crosses for each of the 7 traits. This is called a monohybrid cross. It follows one trait, and the self-crossed plants are called hybrids. In looking at a monohybrid cross, when Mendel crossed true breeding tall plants with short plants, the next generation plants were all tall. When he self-crossed the F1 plants, 1/4 of the plants in the next generation, the F2, were short and 3/4 were tall. Of the tall plants in the F2, 1/3 were true breeding and the other 2/3 were not true breeding. He could tell this by crossing the tall plants to short plants to see which bred true. The monohybrid cross is the F1 generation. Mendel's experiments confirmed that hybrids hide one expression of a trait, which reappears when hybrids are self-crossed. And he speculated that each elementin, which he called it, was packaged in a separate gamete. This led to the law of segregation, which was Mendel's idea that the elementin separate in the gametes. If we look at Mendel's first law of segregation, the mutant phenotype is the variant of a gene's expression that arises when the gene undergoes mutation. He observed the events of meiosis. 2 copies of a gene separate with the homologs that carry them when a gamete is produced. And at fertilization, the gametes combine at random. This slide is showing Mendel's first law of gene segregation. During meiosis, homologous pairs of chromosomes and their genes separate and are packaged into separate gametes. At fertilization, gametes combine at random. Green and blue denote different parental origins of the chromosomes. This cross yields offspring of the genotypes uppercase T, which is the homozygous dominant, and uppercase T, lowercase t, which is the heterozygous dominant. This slide shows the 7 different traits that Mendel studied and the results that he obtained from them. This slide represents how genes in the gametes join if they are on different chromosomes by utilizing what is called a Punnett square. And basically, a Punnett square illustrates how alleles combine in offspring. The different types of gametes of one parent are listed along the top of the square, with those of the other parent listed on the left-hand side. Each compartment displays the genotype that results when gametes that correspond to that compartment join. In a test cross, it was shown that a monohybrid cross yields a 1 homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive in a genotypic ratio. 3 tall to 1 short in a phenotypic ratio. And Mendel distinguished the homozygous dominant, the uppercase T, from the heterozygous dominant, uppercase T, lowercase t, tall plants with a test cross. He would cross an individual of unknown genotype with a homozygous recessive individual. This slide shows a test cross where breeding a tall pea plant with a homozygous recessive short plant reveals whether the tall plant is true breeding, homozygous dominant, or non-true breeding, heterozygous dominant. Punnett squares usually indicate only the alleles. Mendel's second law, independent assortment, considers 2 genes on different chromosomes. The inheritance of one does not influence the chance of inheriting the other. 2 genes that are far apart in the same chromosome appear to independently assort, and numerous crossovers take place between them. If we look at this slide on Mendel's second law, independent assortment, we see that the independent assortment of genes carried on different chromosomes results from the random alignment of chromosome pairs during metaphase of meiosis I. An individual of genotype uppercase R, lowercase r, uppercase Y, lowercase y, for example, manufactures 4 types of gametes containing the dominant alleles of both genes and also the recessive alleles of both genes, and a dominant allele of one with a recessive allele of the other. The allele combination depends upon which chromosomes are packaged together in a gamete, and this happens at random. This slide shows plotting a dihybrid cross. A Punnett square can represent the random combinations of gametes produced by the dihybrid individuals, and an underline in a genotype in the F2 generation indicates that either a dominant or a recessive allele is possible. The numbers in the F2 generation are Mendel's experimental data. Test crosses with homozygous recessive plants for both the R and the Y gene revealed the genotypes of the F2 generation, and this is depicted in the 16-box Punnett square. Probability is the likelihood that an event will occur. The product rule refers to the probability of simultaneous independent events, which equals the product of their individual probabilities. It predicts the chance of parents with known genotypes to produce offspring of a particular genotype. For an example, consider the probability of obtaining a plant with wrinkled green peas. Genotype homozygous recessive for both the R and the Y genes from a dihybrid heterozygous dominant for the R and Y genes parents. This slide illustrates the product rule. In this slide, you can see how these tools can be used to predict offspring genotypes and phenotypes in 3 human traits simultaneously. And here, what has been done is we've taken a man who has normal urine, colored eyelids, and normal fingers, with a partner who has red urine after she eats beets and has normal eyelids and short fingers, and the chance that their child will have red urine after eating beets, colored eyelids, and short fingers is 1/8. If you would like any additional information on Mendel and his experiments, you could follow this link. If we look at single gene inheritance, single gene disorders are rare. The phenotypes associated with single genes are influenced by other genes and environmental factors. If we look at eye color, we see that most people have brown eyes. Blue and green eyes are almost exclusively in people of European ancestry. The color of the iris is due to melanin pigments, which come in two forms: the dark brown, black, eumelanin, and the red-yellow, pheomelanin. In the eye, the cells called melanocytes produce melanin, which is stored in structures called melanosomes in the outermost layer of the iris. People differ in the amount of melanin and the number of melanosomes, but have about the same number of melanocytes in their eyes. If we look at the nuances of eye color, light versus dark brown, clear blue versus greenish or hazel, these arise from the distinctive peaks and valleys at the back of the iris. Thicker regions darken the appearance of the pigments, and they make your brown eyes nearly black in some parts and blue eyes closer to purple. The bluest eyes have thin irises with very little pigment. The effect of the iris surface on color is a little like the visual effect of rough textured canvas on paint. If we look at a single gene on chromosome number 15, OCA2, it confers eye color. And it does this by controlling the melanin synthesis. If this gene is missing, albinism results, causing pale skin and red eyes. A recessive allele of this gene confers blue color, and a dominant allele confers brown. But inheritance of eye color is more complicated than this, because near the OCA2 gene is a second gene, HERC2, and this gene controls the expression of the OCA2 gene. A recessive allele of HERC2 abolishes the control over OCA2, and blue eyes result. A person must inherit 2 copies of the recessive allele in HERC2 to have blue eyes. If blue eye color is a disruption of the normal function, why has it persisted? A clue comes from evolution. The HERC2 gene is found in many species, indicating that it is ancient and important because it has persisted. Perhaps mutations in the HERC2 gene arose long ago among the hunter-gatherers in Europe, and the unusual individuals with the pale eyes were, for whatever reason, more desirable as sexual partners. Over time, this sexual selection would have increased the proportion of the population with blue eyes. Other explanations are possible for this. In this slide showing eye color, you see that at least the 2 genes determine eye color in humans, and inheriting 2 particular recessive alleles in HERC2 abolishes the effect of OCA2, causing blue eyes. The 2 genes are near each other on chromosome number 15. If we look at modes of inheritance, there are rules that explain the common patterns of single gene transmission, and the passing of a trait depends on whether the gene is on an autosome or on a sex chromosome, and whether or not the allele is recessive or dominant. And autosomal inheritance can be dominant or it can be recessive. Approximately 62% of the hereditary malformations and 58% of the known hereditary diseases are transmitted in a dominant pattern. These are often the easiest to recognize as inherited disorders because they're capable of striking many members of an affected family. An average of about half the sons and half the daughters of an affected individual are affected. Therefore, males and females are equally affected with this mode of inheritance. Every affected person has an affected parent unless he or she has a new mutation. In looking at the criteria for autosomal dominant disorders, The affected offspring, like the affected parent, will have a 50-50 chance of having an affected child. The normal offspring with no mutant gene will not transmit the gene to his or her offspring. The severity of the symptoms and dominant traits varies more widely than in recessively inherited traits. And we have what's called incomplete penetrance or variable expressivity. We will be talking about this when we talk about the diseases in more detail next week. We have also the phenomenon of anticipation. This occurs because of the wide variability of expression. The hereditary disease appears to manifest itself earlier and more severely in successive generations. This is only seen when the mutant allele is transmitted by the affected father, not when it is transmitted by an affected mother. If we look at pedigree analysis, for researchers, families are tools. The bigger the family, the easier it is to discern modes of inheritance. And pedigrees are symbolic representations of family relationships, of family histories, and the transmission of inherited traits. I know that you've been looking at these in some of your other courses, and we're going to be discussing them in this course too. Genetic counselors can use family histories to build rapport, to motivate health, behavior change, to monitor and anticipate risks over time, and to inform clinical decisions. It is an extremely important tool. It can improve patient care by early identification of increased risk for chronic diseases or cancer, and it can often improve, delay, or even prevent adverse outcomes. And as I mentioned, it can reveal a patterns of inheritance. It provides information to help make decisions about reproduction also. An autosomal dominant pedigree can demonstrate vertical transmission, approximately equal numbers of males and females that are affected, and male-to-male transmission. Here we can see in this diagram autosomal dominant inheritance. When one parent has an autosomal dominant condition and the other does not, each offspring has a 50% probability of inheriting the mutant allele and the condition. This is another way to denote autosomal dominant inheritance, where in this case you have an affected father and an unaffected mother. And as you can see here, you have 4 children and you have a 50% chance of having an affected child, and it doesn't matter which sex it is. The importance of pedigrees today is it helps families identify the risk of transmitting an inherited disease. It's a starting point for identifying and describing or annotating a gene from the human genome sequence. And meticulous family records are helping researchers follow the inheritance of particular genes. Here is an example of an autosomal dominant pedigree. In this case, you can see that it does not skip generations. It can affect both sexes. But as when we start looking at some of the diseases next week, you're going to see that in some cases, because of the variability of expressivity, it isn't always apparent that a specific individual in a generation has a specific disease. In this case, the disease that we're looking at is polydactylia, extra fingers, and or toes. To interpret a pedigree with it that you're looking for autosomal dominant inheritance, there are some problems. Incomplete penetrance of the gene, a new mutation arising, lack of family history, there may be an adoption, there may be a small family, people may have died quite young, and also the fact that today we have smaller family sizes. Some examples of autosomal dominant diseases are Marfan syndrome, neurofibromatosis, Huntington disease, familial hypercholesterolemia, achondroplasia, and some of the cancer genes, such as BRCA1 and BRCA2. This week, you will do a discussion question on a disorder, an autosomal dominant disorder called ectrodactylia, and you will look at a very sad story about an abused and mistreated individual who did some very bad things himself, who was called Grady Stiles. He lived in the 20th century. He was a circus performer, and he was known as the Lobster Boy. So you will complete the discussion, and have a feeling about so many of these diseases are seen in individuals who were abused and had nowhere else to go except to be in circus sideshows. Genetic diseases that are transmitted as autosomal recessive diseases require that the mutant gene be present in a double dose. The affected individual inherits one mutant gene from each parent. Usually the parents are normal clinically or phenotypically, but they are heterozygous carriers, having a normal gene in one chromosome and a mutant allele on the other chromosome of the pair. In looking at the criteria for autosomal recessive traits, males and females can be affected. Affected males and females can transmit the gene unless it causes death before the reproductive age. The trait can skip generations. Parents of an affected individual are heterozygous or have the trait. It's more likely to occur in families with consanguinity. We will discuss consanguinity in just a moment. This slide just shows you the two heterozygous parents and how they could have children that either are unaffected carriers. That happens half of the time, 2 out of 4 times. 1 out of 4 statistically would be that the child would not have the disease at all and would have both dominant alleles. And then for the 2 recessive alleles, 1 out of 4 times, and that child would be affected. If you look at the next slide, this slide just shows some examples of some autosomal recessive diseases that we'll be looking at next week. Sickle cell anemia, cystic fibrosis, Tay-Sachs disease, and PKU, or phenylketonuria. In this slide, you see a typical autosomal recessive pedigree, and in this case, the affected individual has albinism, which is a deficiency in melanin production, and both parents designated as number 2 and number 3 on the pedigree are carriers in the second generation, number 2 and number 3, I'm sorry, and they are the carriers. Consanguinity is a term used to describe the relationship between couples who share at least one common ancestor. The term incest refers to relationships between first-degree relatives, but may include relationships between a grandparent to grandchild, or uncle to niece, or aunt to nephew. In the United States, there is a significant stigma associated with consanguineous marriage. Many cultures, particularly in the Middle East, Asia, and Africa, prefer marriages between first cousins, uncles and nieces, or double first cousins. Therefore, in some parts of the world, 20 to 60% of all marriages are between close biological relatives. Chromosome anomalies do not occur more commonly in the offspring of consanguineous unions. If we look at third-degree relatives, we look at the risk above the general population, 1.7 to 2.8% for significant congenital defects, and it is 4.4% for pre-reproductive mortality. This slide shows the consanguineous relationships and the proportion of shared genes. If we look at first-degree relatives, parent to child, siblings, half-siblings, it's a half. If we look at second degree, it is 1/4. Third degree, 1/8. Fourth degree, 1/16, and fifth degree, 1/32. This pedigree shows the marriage of first cousins, which in actuality is forbidden and is illegal in many states in the United States, including Connecticut and Massachusetts. If you look at the double line between the female in number 2 here and the male number 3, this double line here denotes that it is a consanguineous. Also, they are carriers for an autosomal recessive disease. Both of them are. We will be talking about working out the probabilities next week. My first job as a genetic counselor was when I was 24 years old, and I went to work at St. Christopher's Hospital for Children in Philadelphia. And one of the groups of individuals that we had coming into our endocrine clinic there was an endocrine and genetics joint clinic, and actually Dr. DeGeorge was the individual who was in charge of that clinic, and we were able to work with the Amish. This was a very difficult thing for me at that early age because many of these children were dying, and the parents felt and would tell the genetic counselors and would tell the other staff that this was God's way, and they kept on having more and more children. And it was a little difficult for me at that young age to understand that. So one of the things when you're looking at the Amish is that they exhibit certain rare diseases that you don't find in other populations. The main disease that we were looking at with these patients was maple syrup urine disease. And some of the other diseases that are exhibited in the Amish population are an Ellis Van Creveld syndrome, glutaric aciduria, Ziegler-Nager syndrome. And some of the illnesses that are found in the Amish are unique to the communities. They aren't found anywhere, and such, for example, Troyer syndrome and Amish lethal microcephalia. And we also see in the Amish, because of the consanguinity, that they exhibit diseases at a higher rate, a higher incidence than the general public. And we see this in muscular dystrophy, deafness, as well as in cystic fibrosis. If we look at why the Amish suffer genetic problems, we can look at some factors. The first factor is small foundling groups. Individual Amish communities typically originate from relatively few families. Almost all of the Amish in Lancaster County, for example, originated from a few dozen 18th century immigrant families. If we look at the factor of genetic isolation, converts to the Amish have been very rare, and Amish only marry other Amish. This practice of endogamy with few new bloodlines entering the population has led to limited variation of genes. So by taking these two factors, the small founding groups and the genetic isolation, we can create what is referred to as the founder effect. And the founder effect means that the genetic variation is lost over time due to a limited number of founding individuals. Certain recessive traits found in a small founding population are amplified in future generations. This is actually an Amish pedigree showing some individuals who were affected with maple syrup urine disease. And maple syrup urine disease is one disease that is actually part of the newborn screening diseases throughout the United States at this time. If you would like to look at some more information about the Amish, I've included in here an article which is very good. It's and Dr. Victor McCusick is one of the individuals who is in the authors. And then also there's a little YouTube on some of the culture. It's a fascinating culture. In looking at X-linked traits, they're patterns of expression on the X chromosome, and they differ in females and males. Females that have X-linked traits are passed like autosomal traits. Males, a single copy of an X-linked allele causes expression of the trait or illness. And a human male is considered hemizygous for X-linked traits. In X-linked recessive diseases, the affected males are able to transmit the mutant gene to all of their daughters who will be carriers and to none of their sons. Affected sons are then able to transmit the mutant gene to all of their daughters. The incidence of the trait is much higher in males than in females. The trait is passed from an affected man through all of his daughters who are carriers to half of their sons. The trait may be transmitted through a series of carrier females. If so, the affected males in a kindred are related to one another through females. These are some examples of X-linked recessive traits. Ichthyosis, which is where you have an enzyme deficiency which blocks removal of cholesterol from skin cells. There will be a diagram of that or picture of that in just a moment. Color blindness, and hemophilia, which is a disorder of blood clotting. This slide is showing X-linked recessive with the carrier mother and how the genes are passed on. This slide shows part of an extensive pedigree for an X-linked recessive disease, the blood clotting disease, hemophilia B. It is also known as Christmas disease and factor IX deficiency. If you look at the top at part A, it depicts the combination of pedigree symbols and a Punnett square to trace transmission of the trait. Dominant and recessive alleles are indicated by superscripts to the X and Y chromosomes. Part B shows how the mutant allele arose in one of Queen Victoria's X chromosomes and then passed to other members of the royal families of England, Germany, Spain, and Russia. The Queen either had a new mutation or she inherited it. Either way, she passed the mutation to carrier daughters and one mildly affected son. For many years, historians assumed that the royal families of Europe suffered from the more common form of hemophilia, type A. However, researchers in 2009 tested bits of bone from Tsarina Alexandra and Crown Prince Alexei for the wild type and mutant alleles for the genes that cause hemophilia A as well as B. Both genes are on the X chromosome. The result was that Mother Alexandra was a carrier of the rarer hemophilia B, and her son Crown Prince Alexa had the disease, which had been known from documents describing his severe bleeding episodes. The transmission pattern of hemophilia B is consistent with the criteria for an X-linked recessive trait. A daughter can inherit an X-linked recessive disease or trait if her father is affected and her mother is a carrier, because the daughter inherits one mutation-bearing X chromosome from each parent. Without a biochemical test, though, an unaffected woman would not know she's a carrier for an X-linked recessive unless she has an affected son. A woman whose brother has hemophilia B has a 1 in 2 risk of being a carrier. Both her parents are healthy, but her mother must be a carrier because her brother is affected. Her risk is the chance that she has inherited the X chromosome bearing the hemophilia allele from her mother. The chance of a woman conceiving a son is 1 in 2, and that of the son inheriting hemophilia is 1 in 2. Using the product rule, the risk that she is a carrier and will have a son with hemophilia out of all the possible children she can conceive is 1/2 times 1/2 times 1/2, which equals 1/8. This X-linked recessive disease is not lethal, and a man may be healthy enough to transmit it to his offspring. If you look at the pedigree, which is on the leg of the individual, and consider the small family depicted in this pedigree. A middle-aged man who had rough, brown, scaly skin did not realize his condition was inherited until his daughter had a son. By 1 year of age, the boy's skin resembled his grandfather's. In this condition, which is called ichthyosis, an enzyme deficiency blocks the removal of cholesterol from skin cells. The upper skin layer cannot peel off as it normally does and appears brown and scaly. A test of the daughter's skin cells revealed that she produced half the normal amount of the enzyme, indicating that she is indeed a carrier. In the case of X-linked dominant diseases, they affect both males and females. Females are affected twice as frequently as males. An affected female transmits the disorder to half of her sons and half of her daughters. It has much more severe effects in males. There's a high rates of miscarriage due to early lethality in males, and it's passed from male to all daughters, but to no sons. Some of the diseases that are X-linked dominant are vitamin D resistant rickets, X-linked dominant retinitis pigmentosa, incontinentia pigmentosa, and X-linked dominant deafness. This is a diagram showing you how the dominant X gene is passed on. You see that you only needed to have the one X to affect the daughter and the son here. And this is an example of an X-linked dominant disorder. This is congenital generalized hypertrichosis. These poor children have been called, this disease has been called the Wolfman syndrome. And if you take a look here, you can see the inheritance pattern. In Y-linked disorders, the genes on the Y chromosomes are said to be Y-linked. Y-linked traits are very rare. They're transmitted from male to male, and a female does not have a Y chromosome. And currently, we've identified Y-linked traits that involve infertility and are not transmitted.